Hi, good afternoon, everybody. Welcome to the e-colloquium. We have a very exciting program today by Professor Gio Travasso. Gio has many hats. He is an associate professor of our department, the director of laboratory for translational engineering. He is also a practicing clinician, a gastroenterologist at the Brigham and Women's Hospital, associate member of the Broad Institute. He is internationally recognized as a pioneering work at the intersection of engineering, medicine, and device development with a focus on drug delivery, ingestibles and implantable systems, and real-time physiological monitoring technologies. Gio has authored hundreds of publications in leading journals. His research has repeatedly translated into human studies and commercial ventures. He has co-founded many companies, including collectively in raising significant venture capitals and advancing new treatment that make it to the clinics. He is also a principal investigator, one of the biggest outreach program with a total funding about $65 million. I'm sure it's a very taxing management task. As an education and mentor, Gio students have achieved many successes, including consecutive years of recognition as finalists in the Collegiate Inventor Competition, underscoring his lab's culture of innovation and translation. And he also have been recognized for many national and international honors, including elected as fellows in the National Academy of Inventors, American Institute for Medicine and Biological Engineering, and the Control Release Society. He is also the honorary foreign member of the Medical Academy in Belgium. Among Gio's many contributions, I think we will hear some very exciting talk today. And I think I should stop and leave the floor to Gio.
Thank you so much, Peter, and thank you all for joining. And it's an incredible honor and pleasure to be here with you. And I was actually looking back at my last colloquium, and it actually took place almost 3 years to the day. It's 36 months minus 4 days. And I was reflecting on everything we've done in sort of these past 3 years. And so what I've tried to do is really assemble a set of slides that try and capture what we've been doing mainly in the GI tract over these 36 months. And I've selected a few vignettes, not all of them, but a few to give you a flavor of the type of work that we do. I recognize the breadth of expertise in this room, so I will try and keep this at a general level, but I will hang around. I'm happy to chat more after. I have the evening free, so if you want to chat more, I will be available. I have some demos here on the table, and so happy to dive into a lot more detail. These are my disclosures. I am an employee and or an affiliate of MIT, the Brigham, and the Broad. I am the co-inventor of several technologies that underpin the work that I will be presenting, and I do receive some consulting fees and or stock options from several companies. These are the ones that are related to some of the work that I'll be presenting today. Now, before I dive in, one of the things I wanted to share with you and really take an opportunity to share is really my background and sort of where I come from and what brought me here to really standing in front of you. I think some of you who know me and have seen some of my presentations, I like to sort of reflect on this to give you at least a sense of my own journey in what brought me to this point. And so a Peruvian, so that's number one. My father met a Nicaraguan, that's number two in England. So I was born in England. I haven't kept the accent, but I was born in Cambridge, UK. And then we moved when I was quite young, around 4, back to Peru. And we lived there for about 9 years. For those of you who are history aficionados, this was sort of a turbulent time in Peru with the Shining Path and lots of other instability. And my parents emigrated. So they were able to get visas to Canada and restarted their lives. And so that's number 4. And I went to high school in Canada, in Toronto. And then I left again. I left for university. And I was an undergrad in Cambridge at Trinity College, University of Cambridge, and I went directly into medicine. And outside of North America, it's something that you can do. And I really enjoyed it, but I always loved research, and I had been exposed to that during at the end of sort of my high school time. So I kept going back every summer. And then I actually interrupted my medical degree with a PhD. That's number 6. I came to the US. I went to Johns Hopkins, and I developed some early tests for colon cancer. I trained in molecular biology, and I worked with someone called Bert Vogelstein down at Johns Hopkins, and those tests eventually were licensed to a small company called Exact Sciences, and maybe some of you have seen some of the ads. There's now a product called Cologuard that took a few years to sort of get approved, but now is broadly available. I then went back to England to finish my medical training, and came over here to the US. And I went and I trained as an internist and as a gastroenterologist at the Brigham and subsequently at MGH. I switched fields into more sort of biomedical engineering, material science, biomedical device development, and I did my postdoctoral fellowship work with Bob Langer, and then initially transitioned to a faculty position at Harvard, and then was fortunate enough to join you here just over 6 years ago. And but during this time, I met an Argentinian, and that's 9, and that's my wife, who is an endocrinologist and an obesity expert, and we had 3 little Americans, which is number 10. And they are here, and I took this picture right before the pandemic, and I took it at the Bodies exhibit at the Science Museum, and I always like to include it to remind everybody the part of the body that we tend to spend a lot of time working in, but also thinking about very deeply, to give you a sense of the size, the dimensions, and which themselves would pose sort of major challenges, but also opportunities to help people. And on the right is generally how I spend my time here in our department, developing new systems to hopefully help people live the best lives that they can. I still practice, usually in between the semesters, so the Brigham has very kindly accommodated me now for many years, and I usually have a couple of weeks of inpatient service time in January, right after New Year's. So what I'd like to try and cover with you are vignettes that fall under 3 general themes, and they're all interrelated. But it's really the engineering of materials for health, the engineering of medicines for health, and the engineering of machines or devices also, obviously, to maximize health. And so with respect to the materials, this is work out of my lab that was initially led by America Tanay, an extremely talented postdoctoral scientist who's now an assistant professor in Minnesota, together with a collaborator, Jan-Ling B, over in Iowa. And then also with James Byrne, who is another postdoctoral fellow. He was a radiation oncologist working in my lab. And the challenge that we set out to tackle is a challenge that I, as a gastroenterologist, tend to see and get referred at. And it's one where you probably have heard of radiation being applied to treat cancer. The challenge often with radiation is that it can injure the normal or healthy tissue around the cancer in addition to helping treat the tumor. And what can happen is that injury can limit our capacity to help the patient, because you can either have very severe inflammation or injury, for example, in the mouth or in the rectum. And so what we said or wonder was, well, maybe we can start to protect some of those normal cells, and maybe we can take a lesson from this organism. And this multicellular organism is a tardigrade or water bear, which many groups around the world have studied because of its incredible capacity to really support itself and remain viable through extreme conditions. And we were very interested in this particular aspect, its ability to actually withstand ionizing radiation. And actually the mechanism by which this was done was described previously just a few years ago, is a protein called DSUP, or damage suppressor protein, that happens to bind a very specific histone protein that is interacting with DNA. So what we wondered was, perhaps we can deliver that specific protein and have that material be produced locally in the normal cells to help protect those cells during radiation therapy. So this was our vision, that perhaps we could help patients, for example, receiving radiation for cancers in the mouth or in the upper oral cavity, but also patients who may suffer otherwise with what we refer to as proctitis or radiation proctitis, which is an adverse effect associated with the treatment of prostate cancer as well as cervical cancer. And so we developed a set of materials, and for those of you who are focused in sort of the field of nanoparticles, We looked at hybrid nanoparticles, including polybeta amino esters that are combined with different lipids. And what we identified were certain lipid nanoparticle formulations that could very effectively enhance the delivery initially of a model reporter, mRNA. And then we said, okay, well, if we take that DSUP mRNA, does it protect the cells from radiation? And we tested this initially in a few cell lines, but specifically in an oral cell line and in a colon cancer cell line. And we said, yes, what we see is that there are essentially more colonies, there's greater viability associated with the delivery of this DSUP. So our hypothesis at least held. And then we said, okay, if we start to apply this in, for example, the buccal cavity of a rodent, do we see less death after radiation? And so in this case, there are certain markers that basically tell us that there's less death. And so indeed, we observed less death. And then the next thing that we wanted to ensure was that we weren't placing a shield and preventing our ability to treat the tumor. So we also looked at our ability to essentially characterize the tumor over time. And indeed, it didn't really affect our ability to treat the tumor. Now, shifting gears to another form of extremophile, and this is work carried out by a very talented postdoctoral scientist in our group who's now transitioned over as a junior faculty member over at BU, Miguel Jimenez. This work was funded by NASA and DARPA. And a few years ago, we started to ask the question with respect to how are probiotics being dosed in the field, how are synthetic biotics being dosed in clinical trials, and what we observed was a pretty broad range of ways in which these new medicines were being dosed. And they could come in the form of synthetic biotics, and certainly there are experts here at MIT that sort of modify bacteria to produce different proteins, or sometimes we mix and match different probiotics, or sometimes we administer stool or complex mixtures, and sometimes they're administered in capsules or enemas. And what we could observe is that the dosage or the frequency of dosing could vary tremendously. And so we said, well, that highlights a concern, which is maybe the material or the bacteria are not viable. So what Miguel did is he went to the local CVS and bought every probiotic that was available on the shelf and asked a very simple question, and that is, how much is actually in the bottle versus what is reported on the label? And there is a discrepancy. And we found that really across the board, that delta did vary depending on the specific type. But we said, maybe material science and through some engineering here on the formulation, we might be able to stabilize those probiotics. So we took what is referred to as the generally regarded as safe list of materials from the FDA and mixed and matched them initially with 3 bacteria and a yeast and put them through a very stressful process. And this is lyophilization. And so freeze drying generally would render many bacteria dead. But what we started to observe, and you can see there in the upper right, is that some of the ingredients enhance the viability of that bacteria. And so we continued to characterize this by leaving essentially these mixtures on the shelf, initially at room temperature, and waiting a month. And eventually we started to tease out which ingredients could maintain the stability out for a month. We also started to characterize whether there was overlap between the ingredients that stabilize one bacteria versus another, and started to understand sort of that universe of sort of potential materials that could help stabilize some of the viability of these live biotherapeutics. Now we focused in on one of the bacteria called E. coli nissl. That is a bacteria that's often used as a therapeutic in traveler's diarrhea, irritable bowel syndrome, and including also inflammatory bowel disease. And we further sort of matured that mixture, that set of materials, through adding now a second component and stressing that bacteria further by holding it at 37 degrees for extended periods. We also compared its performance to the commercially available formulation. And what we found was that we could enhance its viability tremendously. And so I want to focus you on the panel on the right. Our best formulation we refer to as formulation D, which is in green. The blue, which is the maltodextrin sort of labeled there, is actually the commercially available formulation. Essentially is non-viable within a couple of days, whereas ours retains significant viability well beyond 6 months at 37 degrees. Now, we're very interested in really thinking about how we take these systems to help people and how we manufacture them into tablets. Now, taking bacteria and compressing them under significant loads to create tablets is a fairly stressful process for this live biotherapeutic, as is coating with solvents. And that's actually what happens when one is making a tablet. And so there are processes that are referred to as milling the powder and wet granulation. Wet granulation is done with isopropyl alcohol, and the actual tableting and coating processes. And so these combinations of significant pressure as well as exposure to solvents are generally very stressful. But what we found was that our formulations could really remain stable. Now, as I mentioned to you earlier, this was work that was being funded by NASA. And so we were interested whether these formulations could also help protect from ionizing radiation. And indeed, we did see some improvements in the viability. Then moreover, with NASA, one of the organisms we were looking at was actually a symbiont of plants to really enhance the production of nodules in the root structures of plants to facilitate nitrogen fixation. And so in that case, what we did was actually we took those bacteria and we kept them at 50 degrees Celsius to try and model a stressful situation. And we noticed and observed that they could still induce those nodules. Now, with respect, now back sort of to the health side, we also apply these in a pig model of traveler's diarrhea. This is a Shigella model, and showed that we could start to reduce the Shigella in the intestinal loop model. Now, let me shift gears a little bit, and this is work that we did with the Gates Foundation and also supported by a K9N award to a very talented postdoctoral scientist in our group, Gary Liu, who's soon to be moving and joining the faculty over at Georgia Tech in their BME department, where we wanted to develop systems that facilitated dosing in children. Children often struggle to ingest solids, solid capsules, solid tablets. And so we wondered if we might be able to generate a phase transitioning gel that could hold a whole, broad array of therapeutics. And, the analogy or the similarity that Gary likes to hone in on is sort of the Terminator model as far as, changing or phase changing its shape, right? And so what we did was we took essentially a two-part solution where we have a cross-linker, and which is calcium essentially with dithioetridol combined, and then followed by a second solution where we have a polymer that's loaded with the drug. And essentially we have gel formation or gelling occurring, and we have the formation of a tough hydrogel in the stomach of a pig. Just to show you, we added in this case a contrast agent, and these are x-rays of those gels in the pigs. Now, we demonstrated we could deliver small molecules, but importantly, we wanted to really test the limits of our system, and we looked at the delivery of enzymes. We focused initially on lactase as a model, but also on probiotics. So generally, enzymes and probiotics, if they're not coated, they're unable to stay in the stomach for a prolonged period because of the harsh environment. And what we demonstrated in rodents, initially with the enzyme, but finally in pigs, but then also in pigs for the probiotic, was the ability to maintain the viability and activity of those sensitive therapeutics. Now, with respect to hydrogels and this concept of keeping things in the GI tract, it's a general theme, I would say, in our group because it's one of the ways in which we can facilitate dosing for prolonged periods. And so Bin Ying is another postdoctoral scientist in my group, and I should add that he is just about to start as an assistant professor at UT Southwestern. I know he just traveled a couple of days ago there. Developed a system that now uses electroadhesion to facilitate that interaction. And so in this case, what we have is a gel that has polycations, and we can apply essentially a current and then facilitate the interdigitating of those polycations. And also, essentially what we have is interdigitation with the deeper proteins, and that gives us a longer sort of adhesive process that facilitates essentially adhesion out to 30 days. Just to give you an up-close and personal view, this is a picture in the stomach of a pig. And just to show you that it's really adhesion, we have two devices, one on both sides of the stomach. So it indeed isn't moving. So you're seeing one there at the top, and then you'll see the one at the bottom in a second. And then I should add on the upper right corner, one of the things we looked at was delivery of drugs, and specifically in this case a steroid, because we're interested in modulating inflammation, in particular, for example, inflammatory bowel disease. Now, I'm going to shift into sort of the medicines or the engineering of medicines. And the next body of work was really carried out by two very talented postdoctoral scientists in our group, Avik Som, who's an interventional radiologist, and this was during his training at the MGH, and Jan-Jörg Rosenboom, who's a postdoc who has now transitioned to his own startup over at the Engine, and it was in collaboration with Umar Mahmoud over at MGH. And this work has continued. It was awarded a Deshpande grant and has continued in the form of a new company called ABSCO, and also in collaboration with Eric, also at the MGH. And the fundamental concept or observation that we aim to tackle here was something that was described back in 1953. I should say where the label was described in 1953 by R.H. Moll. And I just highlighted here, I took an excerpt from the paper, is that the observation is as follows, is the observation that there's an effect at a distance. And this is called the abscopal effect. So let me just explain. So sometimes if you have a tumor on this arm and another tumor on this arm, it has been observed that in certain situations, if you treat the tumor on one side, the other one also disappears. And what is happening, and this is something that has been delineated and clarified over the last sort of several decades, is essentially there is immunologic training that's ongoing, that ends up essentially facilitating the clearance of the second tumor. And there's a set of drugs that can help stimulate that training. And so one drug that exists is called the imiquimod. It's a TLR7 agonist. For those of you in the field, the challenge sometimes is that if we try and inject that drug, it will quickly leave the tumor. So it doesn't have enough time essentially to provide the signaling required to facilitate the training. And so what we wondered was, perhaps we can address this challenge by engineering the material. So we use the triblockal polymer PLGA-PEG-PLGA here that phase transitions at body temperature and facilitates essentially the delivery of a depot of the imiquimod. And this, by having this phase transitioning liquid, it also facilitates experts like Avik, who's an interventional radiologist, to inject this in tumors all over the body. And what we showed in rodents initially was that we could enhance essentially that abscopal effect using this system. Interestingly, I should add, what we observed was that the polymer itself also had some potential contribution on the abscopal effect, and that it was somehow enhancing also the immunologic response. So this journey continues. It's one that just started a couple of years ago. Now, transitioning to another sort of body of work, and this is a big area that it remains ongoing in our lab, and this is led again by two very talented postdoctoral scientists in our group, Alvin Chan, who's now an assistant professor at NTU in Singapore, and Ameya, who I mentioned earlier. And one of the challenges that we've recognized is really related to a set of medications that I think now we're all familiar with, and it's mRNA. And mRNA, I think we're all familiar with it because of the vaccines. The challenge with mRNA is that if you give it in a naked form, it is poorly absorbed, or it gets broken down very quickly. And so you need to deliver it in a vehicle. And the vehicle is a lipid nanoparticle that we're most used to. And so now the challenge with LNPs is that they're composed of different elements. And the traditional LNP is typically composed of 4 elements, and that there's an ionizable lipid, a helper lipid, a PEG lipid, and a cholesterol. And very small variations among these 4 can have a profound effect on the actual delivery efficiency. And so what they did is develop a transformer-based model where we did a large number of in vitro experiments, and then were able to train the model with respect to each one of these components to accelerate our ability to predict and identify LNPs that could deliver various payloads to different parts of the body. I'm just going to show you this body of data, which is part of the early characterization, where we took some of the hits, the in silico hits, and compared them to clinical benchmarks in rodents. And what we found was that we could enhance delivery 5 to 40-fold over those clinical benchmarks. Now, this is being applied across a range of different areas, and so certainly that continues. Now, with respect to injection, I'm going to transition briefly outside of the GI tract, just to share this particular body of work, which is certainly a big part of our lab's efforts in developing systems to support long-term drug delivery. This is work that was co-led by Vivian Feig, who's a very talented postdoctoral scientist in our group, who's now assistant professor at Stanford. Sang-Hyun Park, who's a final year PhD student. I should add, he's also a finalist in the Collegiate Inventors Competition, so please do vote for him if you're able to. And Beppe Ravano, who was an ETH master's student with us, who's now back in Zurich. And the challenge that we set out to address is that of really self-administration. So when one looks at the needles that are out there that one might apply for delivery, these are some of them. And what we recognize is that, and we include a grain of rice there just, for frame of reference, is in general, I think if one has to inject themselves, they would prefer to do it with a smaller needle. The challenge is that in order to deliver a depot, so something that is going to release a drug for a prolonged period, it is hard to do that because often we are delivering something that is packaged in a microparticle, for example. This can be done for shorter periods, and certainly the early example I gave with the Miquimod, that was sort of a week-long depot, but if we want multiple months, we typically need sort of solid particles to do that. And so our vision and dream really was, maybe we can figure out a way of applying the unique physical chemical properties of a certain class of drugs and identifying solvents that can work well, keep these in a suspension or fully dissolved, and then have them essentially coalesce in the body. And so that's exactly what we did. And so we basically determined a combination of a solvent that's biocompatible, but also drugs. And we focus on levonorgestrel, which is a contraceptive agent, so that we would have essentially coalescence or self-aggregation of these systems. And so by screening several different essentially drug solvent combinations, we identified biocompatible solvents that have been used or are in use in a variety of different injections that could form these solid, essentially self-aggregating systems. And then honed in on the benzobenzoate formulation, we observed some toxicity with the benzyl alcohol formulation. And so we focused in on the benzobenzoate one, and we delivered these in rats. And what we actually engineered was the ability to modulate the porosity of that solid sphere. And so, for example, if one takes levonorgestrel with phosphate buffer saline, so an aqueous system, what you have essentially is sand that is, it is coalescing locally, but there's a high surface area as reflected by the packing fraction. And so that manifests in higher levels of drug. Now, if we take the levonorgestrel with benzyl benzoate, we get better, more compact self-aggregation. That packing fraction increases, the surface area of interaction decreases, and the drug levels are a little bit lower. We can further decrease that by adding a tiny amount of polymer, and we used polycaprolactone, which is a biocompatible polymer used in other drug delivery systems. So this system continues, and we are applying it to several other drugs at the moment. Now, while we're sort of on the drug delivery sort of journey, let me share this particular project that continues. This is Louis de Ritter, a graduate student in my lab. Aaron Lobes, a very talented research engineer in my lab, and our collaborator, Doug Rubinson, who was my co-resident a long time ago, but is now an oncologist and gastrointestinal cancer expert at the Dana-Farber. And one of the things that I remembered when I was a resident, so when you're sort of in the hospital, you're rounding on patients who are receiving chemo when you're on sort of the oncology service. And one of the things I recognized was that the way that many chemos are dosed is based on weight and body surface area. And several years ago, I wondered, why, right? And this is unbelievable. So in 1916, there was a small study of 9 people, and they derived an equation that essentially, to this day, is the equation that governs how chemo is dosed. And it is purely based on height or weight. Now, our understanding of how drugs are processed, the microbiome impact on drug levels, diurnal variation, of course, has blossomed, but yet we stick with this equation. So what this means, maybe to bring it home a little bit, more concretely, you can take two people, and I'm giving you two examples here, and these two people will receive the exact same amount of medication. So that, at least to us, or to me, never quite made sense. And this is actually borne out in the literature, where we see tremendous variation across individuals, and that can manifest in increased toxicity or underdosing. So we said, well, is there something we might be able to do and come up with to try and develop a closed loop system where we can actually dose and aim for a very specific level? So we developed this system we refer to as the closed loop automated drug infusion regulator, or Claudia. And so using Claudia, what we demonstrated, this is work initially done in rabbits, and we used a very common drug, 5-FU. We selected 5-FU because it is broadly applied across many, many cancers. And so we recognize the potential to really help many people using this technology, but the platform can be applied to many other drugs. But using a closed loop system, we demonstrated our ability to actually reach the therapeutic window in every single animal, as compared to variations that are observed just by using the equation. And we also inserted in, when we were doing all this work, we also modeled interventions of, for example, adding an inhibitor, trying to mimic, for example, someone ingesting a specific drink or food that also might impact, for example, the processing of the metabolism, I should say, of the drug. Now, I want to share a little bit of an update on a technology that we've been working on for over a decade. And this has been an incredible journey. This is just a snapshot of the people that have been involved. There have been over 100 people, but it's been an incredible journey that continues. And this latest, I would say, chapter was led in collaboration with Leslie Citrome from Colombia. And just to maybe just remind you and give a little bit of background, one of the fundamental observations out there that we recognized over a decade ago is that if one administers drugs more infrequently, the patient is more likely to actually continue and stick with the medication. And I think this is obvious if you consider having to take a medication once a day versus 4 times a day. But it's interesting that you can actually stretch that interval, and that observation holds. And that's what I show you on the right is a trial out of Japan with medications that are available in daily, weekly, monthly, and actually even yearly dosing schedules for osteoporosis. And so with this in mind, several years ago, we sort of envisioned this particular concept. Could we develop orally metered dosing systems? And some of those early concepts were captured here by Matt Kaplan in The Economist, talking about some of that early work. And so we did it. We developed a capsule that is able to essentially reach the stomach, open, and because its mechanical properties, is able to withstand the compressive forces of the GI tract, and also because of its dimensions, is able to stay essentially in the stomach and slowly release drug. And we can control the interval during which that process is occurring, because we can include very specific materials that we engineer to break down over a week, 2 weeks, or whatever the desired period is. We applied this academically before to different situations: malaria, HIV, and even a once-a-month oral contraceptive. But the biggest, I would say, the latest sort of result really came in a paper we co-authored just a few months ago, where we demonstrated phase 3. This is a human clinical trial, repeated dosing in patients suffering from schizophrenia, a condition where the condition impairs adherence. And what we demonstrated here was essentially when we compare daily dosing of the therapeutic, that's in the orange on the left, what we demonstrated was that in repeated dosing, the levels of the drug remain within the bounds of that daily dosing over 5 consecutive weeks. Now, transitioning to the machines or device section, I'm going to start with this particular journey, which is really around the development of ingestible sensors and really helping characterize, developing tools that can characterize gastric electrophysiology. And this body of work was led by 2 incredible engineers. Adam Gerlach is a current graduate student in our lab. And I should add, just like Sung Hyun, who was a finalist, Adam was a finalist last year in the Collegiate Inventors Competition, and Sean Yu, a very talented postdoctoral scientist who's at a local biotech. And what we recognize is that, over 100 years ago, people started thinking about trying to essentially characterize the GI tract, very specifically the electrical signals from the GI tract. So certainly there was a tremendous body of work looking at EKG and its application to really characterize what was happening in the heart, but also there were efforts to really characterize other electrical signals. And so there was information, and certainly this field has moved on, and there have been some very nice publications by other groups looking at essentially surface electrodes and characterizing some of the signaling. But what we recognize is that there's tremendous attenuation of the signals. So we said, maybe we can create capsules that can actually give us direct information from this tissue. And so in these sort of images, you can see sort of those early prototypes of these capsules that have an array of sensors that are able to unravel and capture these signals from the local tissue. So we apply these systems, and we refer to it as MyGut, which stands for multimodal electrophysiology via ingestible gastric untethered tracking. And what we're able to collect is a rich body of data, and using a range of different bandpass filters, we're able to pull data that provides information on cardiac function, respiratory function, but of course, what we were interested also is the gastrointestinal function. And we looked at it both in sedated and also unsedated models. And what we demonstrated was our ability to also be able to capture, for example, feeding events, walking or sleeping. And of course, additionally, we observed sort of, and in our control experiments, the ability to modulate the contractility of the GI tract using various drugs, whether it's a prokinetic like azithromycin that has motilin stimulation effects, or in the setting of barbiturate dosing. Now, shifting gears a little bit, but still sort of in the area of electromechanical aspects, Shreya Srinivasan is a postdoc in our group, working closely with Keir Byrne, who's here at the back. Also a very talented postdoctoral scientist who really worked together to characterize a fascinating phenomena, which takes root back almost 50 years, or just over 50 years ago. There were these incredible experiments that recognize how vibration of muscles could create an illusory sense of movement. And essentially, the position of the arm, for example, in these experiments, could be affected by the presence or absence of that vibration. And so this stimulated inquiry into whether we might be able to simulate an illusory sense of fullness. And so the vision was, could we develop a capsule that stimulates the gastric cavity and create such a response? And so we conducted a set of experiments where we took a pig and we inflated a balloon. We also actually captured the signals from the celiac vagal branches. And what we started to characterize was essentially the signaling that's associated with that balloon, and compared it to vibration across a range of frequencies. And we found frequencies that seemed to align with essentially the signals that we were getting from the balloon expansion. And then beyond that, started to characterize some of the hormones that start to inform whether there is a picture emerging consistent with satiety or fullness. And this was supportive. But finally, what we were able to do is take pigs and essentially record their behavior to try and capture whether their feeding was reduced. And what we were able to quantify from these early studies was actually our ability to modulate that feeding, or how much food was left in the hopper. Now, along the lines of ingestible devices and satiety, a few years ago, we started on this journey together with Jimmy and Khalil. Jimmy was a graduate student. He just graduated a few months ago. A very talented graduate student, I should say, also similar to Sang-Hyun and Adam. He was also finalist at the Collegiate Inventors Competition. And Khalil Rahmati was a postdoc in our lab. He transitioned to a faculty position a couple years ago at NYU. And the fundamental observation that really led to this body of work comes from this. And this is a pacemaker, essentially, that is sitting in the stomach. It's the Medtronic Entera system. I believe Medtronic recently sold it to another company. But what it is used for, applied for, is a condition that's referred to as gastroparesis. Gastroparesis is when the stomach isn't quite working. It's moving slowly. And the thought here is that we might be able to pace the stomach. What I can share with you as a clinician here, at least in Boston, we're unable to actually request this to be placed. And the reason is that actually the prokinetic, or that stimulation, doesn't seem to be that effective. But if one looks at the clinical trials and the retrospective analyses of people who have received this, patients report 10 years after implantation that they actually feel better. And by that I mean reduction in symptoms of nausea and vomiting. And so this made us wonder that perhaps what is happening is that something is being stimulated. And so we re-engineered a tool that gastroenterologists like myself use, which is called the Gold Probe. We typically use it for cautery. So that's a tool that we typically use to stop bleeding and ulcers. But we re-engineered so we could actually now stimulate under parameters that match the Entera system. And what we observed was that we could induce ghrelin, which is the hunger hormone, which interestingly also modulates nausea. And so we fine-tuned some of the parameters. And one of the challenges that we started to think about was, well, if we try and stimulate in the stomach, big challenge that there are a lot of puddles in the stomach. And so we would be worried about parasitic losses of that stimulating event. And so we were inspired by this animal from down under that has essentially this capillary structure in its skin. And we said, maybe what we can do is create capsules that have these channels that can help wick away the fluid while maintaining contact with the stimulating electrodes. And that's exactly what we did. And we were able to package the parameters inside of a capsule. And so we had a custom PCB, and we're able to essentially put this together and give it to animals, and we're able to recreate that stimulating event. Now, while we're sort of on the ingestible device side, this is work that actually my first graduate student in MECI, Graham Arick, led in collaboration with folks at Novo Nordisk. And it really tackles a fundamental challenge in that I think most people, if they have to take medication, they would prefer to take it with the object on the right over the one on the left. Now, this fundamental observation isn't new, and I just want to give a little bit of background and history. And so over 100 years ago, insulin was first described up at the University of Toronto by Banting, Bass, MacLeod, and their team. And the first injection of insulin was given in 1922. At the beginning, it didn't quite work, and it took further processing and purification, but eventually it did. Now, interestingly, within months of that first event, the idea of oral delivery of insulin started. And there was a paper the paper came out in 1923, but the experiments actually started in 1922, that describes an experiment where insulin is mixed with a weak alcoholic solution to try and look at whether one could get absorption. And what's remarkable about this paper is actually there was one case of the 6 that showed a decrease in the glucose level, so insulin was being absorbed. Now, what I can share with you is that many have sort of chased this particular challenge to a range of effects, but the challenge is that insulin, like other proteins that we ingest for nutrition, are broken down or are too big because the hydrodynamic radius is such that it cannot travel through the GI tract. And so this is the fundamental challenge that we set out to address and have worked on now for several years, but really inspired by cephalopods and the jets that cephalopods use for translation movement in their medium, we thought maybe we could create devices that could jet the therapeutic into the mucosa. So we developed a family of devices and looked at the delivery of biologics. So that includes insulin, RNA, monoclonal antibodies, so even molecules that are much bigger and harder to deliver in the GI tract. And we were able and were successful And just to give you a flavor and a sense of what this looks like, so these are some of our prototypes. This is in vitro, but you can see sort of those jets actually delivering through the medium here. And what we did as part of this body of work was we characterized the fundamental, essentially, pressure and also the volume of the depots that we could generate everywhere in the GI tract of a large mammal. This is work in a pig. And as I mentioned, we characterized several different drugs with respect to our ability to deliver them. I show here curves for both the GLP-1 analog and also siRNA. One thing I want to just highlight is that the concept of bioavailability essentially is the fraction of drug that actually reaches the blood, typically for an injection, like a subcutaneous injection of insulin. That bioavailability is somewhere between 60 to 80%. Oral bioavailability of insulin is, I believe, 0.001 or 2, so it's very low. What we're able to achieve with these systems is close to subcutaneous administration, so upwards of 50%. Now, one of the things that I've mentioned to you now a few times is the fundamental challenge of trying to adhere to the GI tract. And the reason that we're so interested in that concept is because it enables our ability to deliver drugs for a prolonged period. It enables our ability to sense and measure a range of analytes. And we have certain ways of doing this for short periods of time, in some instances for longer periods of time with chemicals and other devices. And recently, this is work that's been led by a very talented postdoctoral scientist in our group, Troy Kang. And Troy is really focused on a new approach. And this is a challenge that certainly affects the GI tract, but also affects adhesion to skin, right? Any surface that is wet, that's dynamic, that turns over, remains a challenge with respect to adhesion. And what we wondered was, are there lessons in nature that might inform fundamental engineering processes? And so with respect to the GI tract, we were thinking about shear because of the food and liquid that's ingested. And so we turned to remora, or the suckerfish. And we asked a fairly simple question. It took some time, but we said, what or how have remora evolved to adhere to a broad array of surfaces? And so what you see here is that analysis of essentially how the sucker organ is capable of adhering to a very broad range of substrates, ranging from sharks to turtles. And we were able actually to get remora and study them very closely. And so we studied this particular part of the organ and recognized that the interaction really is across length scales from this large sucker essentially at the edges, and we essentially start to come down into comb-like structures, the lamellae, and those are further sort of subsegmented into these sort of small villi. But we were really interested also in the fundamentals of how these systems interacted with tissue. So we actually were able to study them. We collaborated with colleagues over at Boston College, and were able to record these interacting with various substrates, including synthetic stomach. We're interested in the GI tract, and so we wanted to understand that interaction. And essentially just teasing out sort of, many tens, hundreds of hours of videos, what we see is sort of these fundamental steps of contact, the unfurling, and eventual adhesion. And we used all of this information really to inform this fundamental engineering, where we have, again, that large macro structure, the comb-like system. And in this case, we don't have the dynamic component of the animal, but so we added a shape memory alloy, so this is nickel titanium alloy, nitinol, to really help impart that dynamic component to facilitate the interdigitation of the lamellae with the underlying substructure. And so using these devices, one of the things that we recognized early on was, the fish is able to swim to facilitate that adhesion, but we also recognize that the GI tract has a peristaltic motion. And so I'm showing you here an example of peristalsis, and you have adhesion there of the device to the esophagus. So using these systems, we looked at a broad range of applications from wildlife monitoring. So here we applied it to tilapia and monitor them in a tank for prolonged periods. We were able to keep it actually in place for a few days. And we're actually speaking with the Navy on potentially monitoring some other fish using these systems, because we are able to keep them on for several days without harming the animal. Now, going back to the GI tract, one of the things we were interested in was seeing if we might be able to develop better systems for evaluation of reflux. And reflux is essentially the condition where material from the stomach goes up into the esophagus, and essentially it's extremely uncomfortable, and over time can result in chronic injury, and eventually, in some instances, can lead to esophageal cancer. And so we essentially were able to measure impedance in the pigs and demonstrated our capacity to measure these reflux events. Going back to our fundamental core challenge of long-term drug delivery, we looked at HIV and we looked at antiretroviral delivery over the course of a week. So using these systems on the back, essentially, of the synthetic remora, we're able to place a long-term releasing system capable of releasing an antiretroviral over the course of a week. And the very challenging situation that we looked at was delivery of mRNA. So I shared with you some of the challenges that mRNA delivery carries with it, but we showed successfully our ability to deliver mRNA in the GI tract using these systems. So that sort of hopefully gives you a quick fly-through of some of the things we've been up to. I think one of the joys that I have of being in the department is being a member of the 275 medical device design class. We've also published there, and we certainly, it's, I think, an incredible class. So all the students in the room, please join us and take the class if you haven't. I'm on sabbatical this year, so maybe next year. But I wanted to just share with you some of the highlights from the last 36 months. So just 2 years ago, one of my teams led a project together with the Gates or Global Health Labs, which is Bill Gates's lab that he supports. So Gates Foundation typically gives you money, but Bill also has a lab that actually develops devices. And so they came to us with a challenge, and they wanted a very low-cost, single-use system that could help support amplification for diagnosis. Amplification, by that I mean PCR-based systems for field-based diagnostics. So this body of work actually was published at the IEEE conference that's actually held here. But we also were able to take the team to Seattle, and it was an incredible visit, I think, for everybody. Now, 2 other recent projects over the past few years. One published in Journal of Medical Devices, and the other one they just presented a couple of months ago at EMBS. The first really is to tackle the challenge of visualizing the cervix. And so here the team really developed a new way of expanding essentially the vaginal canal, facilitating visualization of the cervix. And in the second, this team focused on a basic challenge of essentially when someone has a mole that's being evaluated, often it will be cut and then someone has to suture. And so that takes time. And so one of a local dermatologist said, do you have any ideas, or could you help us figure out how to do this more efficiently? So we developed a single-step device that is able to take that biopsy and close the wound. And as I said, this was just presented at the EMBS just a few months ago. So all of these are from 275, so I hope you come join us. Now, I know we're close to time. I just want to thank many people, but first of all, our incredible team. So we're very fortunate to have our team, but this is also their families and friends at our summer party. I'm going to just show you, these are folks from 23, 2024, and then just this past summer, and we celebrated on one of the Boston Islands. But I also want to thank our incredible department. I took this picture after the celebration for my tenure, but there are many more people that weren't in the picture that I wanted to just acknowledge. I think it's an incredible community. And I know I'm almost at time, and I will hang around, but if you have questions that or things that interest you, or interested in collaborating, here's my email. I also want to acknowledge the incredible support from our sponsors. And with that, I thank you for your attention.
Thank you, Gio, for a wonderful talk covering must be 15 areas. And so I'm sure that there are many questions that would arise from the very interesting talk.
Yeah.
Yeah.
Thank you for the wonderful talk. Just a very like general question, I guess specifically regarding the synthetic remora. Like when it adheres to the GI tract, is there any concern of like a negative or harmful foreign body response? And then more generally, are foreign body responses an issue, or foreign body responses to these implants an issue in your devices?
Great question. So with respect, maybe I'll answer it a few different ways. So as a clinician, one of the things that I do in people, if they come and I'm evaluating them and treating them for an ulcer, is I will put an alligator clip, very similar to an alligator clip that you might use in basic electronics. We have, they're called endo clips, but it's the same thing. And they will sit there for up to a year or even longer. Okay, we also, in the case of remora, we're actually not creating any defects. And so it's purely through adhesion, and at least in the preliminary studies we've done in pigs, we haven't seen any injury. So I'm giving you a clinical extreme, but and there's the preliminary data that we have from the early studies. I think that as these projects continue, your question is critical, and in the medical device development sort of process, safety comes first, and that's the kind of question that we would want to understand by studying it further in other species and eventually in people. Yes, Gary.
Going back to one of the earlier parts, you had an acronym LIFT for an in situ two-part hydrogel that formed in the stomach. And the T in there was for tough, I think.
Yes, and I didn't show any of the mechanics
so
there.
I'm immediately curious about how tough and how do I measure that in the stomach?
Yeah, so that was measured all externally. And so, yeah, happy to go over the details. But we didn't measure it in the stomach, we measured it on the bench. And then functionally, we measured it in the stomach with respect to its ability to form the objects and also to withstand passage. In that case, lift is interesting in that it will stay for a day, but it won't stay for multiple days. And so, yeah.
But it is, so roughly?
It's a double network, and I don't remember off the top of my head the parameters, and so I'm happy to follow up, Gareth, and send you the details.
As people thinking about questions, let me ask one. So you have the COMET system of trying to identify the best combination of lipids for the delivery. So just wondering, what is the parameter that you change in training the network? And what is the insight in terms of the different type of lipids? How does it optimize the delivery in that case?
So the parameters that we're changing are the ratios of each of the 4 components. And that is...
For each of the components, does it have different type of lipids?
There's 2 things, exactly. So we vary the specific component and also the various ratios of the individual components. Mm-hmm. Additionally, so that was the base. We actually did a large in vitro campaign screening in 2 cell lines where we made 3,000 different formulations and evaluated essentially the expression of luciferase. And that is essentially what informed the first part of the model. Now, if we add a new component, what we have observed is essentially we can run a much smaller training campaign, generally of 300, so 3 plates, that is able to then achieve the similar predictive capacity as the original 3,000. Mm-hmm. And so what we've done is we've looked at a few things. So we've looked at adding new materials. And so we demonstrated in the original description, the original paper, that, for example, if someone is exploring a new material that they want to try and incorporate for whatever reason inside of an LNP, we can essentially explore that through a limited set. We also looked at varying the destination. So we had looked at in 2 cell lines initially, a dendritic cell line and a melanoma cell line. And so, we're interested in the GI tract. So then we added a training data set around GI lines, and again, we're able to identify hits for GI lines following, again, a much more limited training set in the context of obviously the prior body of training. We also looked at stabilization. So we added a stress to the LNP. So for example, if we take LNPs and hold them at higher temperatures, and then essentially identified formulations that were more stable. So those are the parameters that we've looked at. I mean, I think certainly one can look at other stresses, and then go from there.
Oops, sorry.
Thanks, Gio. That was an amazing talk. I have a question about the nitinol lamellae. How do you remove that afterwards? And is there any risk of corrosion of the nitinol in the acidic environment?
Yeah, no, great question. So the nitinol and all of these devices are just excreted, so we never remove them. I mean, they're small enough that they're excreted. They're compatible with other ingestibles. I think as far as the corrosion, it's not something that we've looked at on extended periods beyond the periods that we've looked at, so about a month. But what we've seen is we will see some corrosion. I mean, some of the things we've talked about are further coatings to try and mitigate that. But yeah, that's sort of where we're at right now. Yes.
Thank you very much for this.
No, thank you for coming.
It's amazing techniques. My question is to translate those techniques to medical practice. Probably the expected challenges, there's the individual's experience, and there's also, I assume, this chemical and there's all the complexity in the GI tract may impose on other challenges to make a reproducible result. So what will be the general strategy to make a medical device that will work in the GI tract and to deliver the drug and this therapeutic outcome in a reproducible manner?
So I guess I'm just trying to understand, so how, the specific question, so what exactly, from a reproducibility perspective, and what aspect are you concerned about?
So one of the challenges might be this in the translational purpose, the variance and the anatomical or this physiological variance can be one of the expected challenges to make a therapeutic purpose. And also, I assume the GI tract has complex chemical environments compared to the other location of the body. That's how we could make it, just control those variability.
Yeah, I mean, the variability is in itself one of the challenges, right? And I think that understanding that variability is critical to inform what variability is acceptable. So I'll give you some examples. The time it takes for an object to transit from your mouth to your stomach can vary from a couple of seconds to tens of minutes, okay? The time it takes for an object from your mouth to your intestine can vary from minutes to many hours. So understanding and having that understanding that those ranges exist among the population start to inform the types of applications for oral drug delivery in general, beyond even what we do, right? So there are ways of trying to mitigate that insofar as, for example, drinking water and standing up, for example, are some of the ways in which even FDA labels actually inform the ingestion of tablets. And so, that your question on how do you reduce variability, it's by studying it in the specific populations, it's by looking at the dosage form, where do you want to deliver it, and then thinking about what range of variability is acceptable. So for some of the approaches we're taking, we are looking at drugs that we want to ensure are delivered on the order of single minutes or even up to 10 minutes. And so we want to focus, for example, on the stomach. And so that jetting system, for example, we know because of its weight and because of its transit time that in general it reaches the stomach within a minute. And so we know that we can have it trigger somewhere between 3 to 6 minutes, and that ensures that a high fraction of people receive that medication. So I think there are many different parameters, and it just depends on your goal. I think if it's a drug, I'll give you another example. So semaglutide, which has gotten a lot of attention in the form of Wegovy, is a drug that has a very long half-life, and it is not acutely needed. And so that's a drug that can be delivered and slowly taken up, and the timing isn't critical, right? Because it will stay in your body for 300 hours. And so there, how you're fitting essentially the delivery component, the dissolution component, and how that's married, with the technologies, I think, starts to vary. So I think your question is very broad. I think I'm just giving you some examples of things that one would think about and consider in drug delivery to try and answer and come up with a strategy that meets the requirements of the population that you're trying to help.
Thank you very much.
You're welcome.
Thank you for the great talk, Gio. I have a questions about like overall ingestible devices. When you think about some clinical translation, how could we remove ingestible devices from patients? And do you have any possible that we can reuse the device to the patients or something?
So in general, I would say reuse would not be something that, I think that would be very difficult from a regulatory perspective because of the infectious risk. And so, just to start backwards. Now, how to remove them? Devices are removed either endoscopically or surgically, or they're purged. And so, there are examples where that has to be done. In general, we're asking someone to take something, we really want to avoid having to go in and remove it. So I would say, I hope that we don't have to be in that situation. And there are tools to actually inform that. So there's something called a patency capsule. And that is a capsule that is essentially made of a dissolvable material, but it has very specific dimensions. And so what we do in patients where we're worried about obstruction or essentially getting stuck, we ask the patient to take that and take an X-ray after a certain amount of time. And then, if everything's working fine, then it should be excreted or a certain position in the colon. If it's not excreted or it's stuck, then we would be careful about giving anything that would follow on, because that will dissolve, but obviously a solid object would not and may manifest in an obstruction. So, we have endoscopy, we have surgery, we have purging, but I would say prevention is typically what we try and do so that we don't have to remove anything.
So, given the time, and we should probably thank Gio for a really wide-ranging talk. Of course, a lot of work in the GI tract and tracking the bioevidence. Thank you very much, Gio.
Thank you.